Our topic is DNA synthesis. DNA stands for deoxyribose nucleic acid. It is a molecule inside organisms that contains the genetic information responsible for their development and functioning. DNA allows this information to be passed from one generation to the next generation. DNA structure, as you can see, DNA has a double helix shape which is like a ladder twisted into a spiral. Each step of the ladder is a pair of nucleotides. Nucleotide is a molecule made up of deoxyribose, a kind of sugar with 5 carbon atoms, a phosphate group made up of phosphorus and oxygen, and nitrogenous base. DNA is made up of 4 types of nucleotides: adenine, thymine, guanine, and cytosine. Adenine and thymine can pair up because they make 2 hydrogen bonds, and cytosine and guanine pair up to make 3 hydrogen bonds. Adenine nucleotide A always pairs with thymine nucleotide G, and guanine G always pairs with cytosine nucleotide C at GC. Double helical nature of DNA. It was discovered by Watson and Crick. The strands of the double helix can separate and serve as templates for the synthesis of daughter strands. It was determined that DNA replicates via a semi-conservative mechanism. Requirements for DNA synthesis. There are 4 basic components required to initiate and propagate DNA synthesis. These are substrates, template, prime, and enzymes. The replication of both parent is a discontinuous process. The strand that is continuously synthesized is called leading strand, while the strand that is discontinuously synthesized is called the lagging strand. Lagging strand synthesis. DNA synthesis require a primer usually made up of RNA ribose nucleic acid. Primases synthesize the ribonucleotide primer ranging from 4 to 12 nucleotides in a length. Only one primer is required for the initial strand synthesis. More complex. Factors involved in DNA synthesis are helicase, which unfolded DNA. Primase synthesize the RNA primers required for initiating leading and lagging strand synthesis. DNA polymerase, which recognize the RNA primers and extends them in 5' to 3' direction. Topoisomerase removes the positive supercoils that form as the fork is unwound by the helicase. RNase H, which removes RNA portions from Okazaki fragments. Ligase, which seals the nicks after filling in the gaps left by DNA polymerase. This is 2 leading strands and lagging strands help in DNA synthesis. This our lecture on the leading strand. DNA synthesis occurs continuously, and lagging strand DNA synthesis restarts many times as the helix, resulting in many short fragments called Okazaki fragments. Basically, these Okazaki fragments are DNA or RNA segments which help in lagging strand to make DNA synthesis. DNA ligase joins the Okazaki fragments together into a single DNA molecule. Thank you.